Thank you very much. It's a great pleasure to be here, and I have really enjoyed my day, particularly talking with several young scientists who I hope, if they were looking for words of wisdom, the one thing I told them was not to take anything I said very seriously. So before I actually give my sort of talk on antibiotics, I want to take a few minutes to talk about the current situation that the UK and by implication the rest of Europe finds itself in. And that is collaboration, scientific collaboration, and how that might go forward. And you know, both the UK government and the EU understand, or seem to understand, that maintaining the closest possible cooperation on research is in everyone's interest. For example, Theresa May's recent speech on science makes the UK position very clear, and Pascal Lamy's report on the future of EU research and the recent launch of the proposals for Horizon Europe are all very positive. But we can't be complacent at this time, because all of our governments need reminding just how crucial research is to a strong future in Europe, and the fact that the closer our cooperation, the more we all gain. Now, UK researchers, first of all, the vast majority of UK scientists wanted to remain in the EU. That's what all the polls show. But they understand that by leaving the EU, we're giving up our right to be part of Horizon Europe. But we hope that our commitment to international cooperation will earn us a very close association. The UK scientific community has been very active in most EU programs. In the ERC, we've contributed a lot to panels, to expert review, and so on. And that's simply, and we're active participants in many other areas of science, for example, in CERN or the ESA. So it's simply an example that, you know, UK science is really collaborative and outward-looking. And in particular, concerning our two academies, you know, the Royal Irish Academy and the Royal Society have had a long history that well predates the EU, and that collaboration continues till today. And one example is the 2018 International Exchanges Cost-Sharing Program, and there are already they're going to enable 2-year high-impact collaborations between researchers based in Ireland and in the UK. And of course, the Royal Society's University Research Fellowships, we, in collaboration with the Science Foundation of Ireland and the Royal Irish Academy, we have sort of been involved in choosing URFs and so on. So we already collaborate on a number of things, and I hope that somehow science doesn't become a casualty of other you know, broader considerations. But actually, I hope that in the end, a sensible overall solution is found to this somewhat difficult political problem. Anyway, having said that, I've deliberately left this on the screen while I was speaking, and even before while you were waiting, because I'm sure you've all figured out what these writers on top and these musicians on the bottom from different countries all have in common. And of course, what they all have in common is they died very young of infectious disease. And we now seem to think that, you know, that belongs to a past era. But the reality is that there are 25,000 deaths a year in just Europe alone from infectious disease, and largely due to antibiotic-resistant bacteria, and often due to MRSA, which is methicillin-resistant Staph aureus. And in fact, the idea that bacteria would become resistant to antibiotics was known a long time ago. In fact, Alexander Fleming actually, when he accepted his Nobel Prize, warned that bacteria would evolve to beat antibiotics. So when you look at the modern era of antibiotics, there were sulfur drugs, which were purely synthetic. Then there were biological antibiotics, of which penicillin was one of the first big ones. And after the discovery of penicillin, there was a systematic search to find other biological antibiotics. And that resulted in a large number of antibiotics, some of which are listed here. And it turned out many of these work by blocking protein production in bacteria. And all life depends on proteins, because proteins not only give us structure and form, but they carry out all the chemical reactions in the cell. And so without proteins, no life can exist. And if you stop bacteria from making proteins, they die. So the question is, how are proteins made, and how do these antibiotics block them? Well, first of all, this is what proteins look like. They're long chains of amino acids, and they can fold up into different shapes. Some of them are filamentous, like collagen, which forms your skin and connective tissue. Others have different shapes. This is a hemoglobin, which carries oxygen in your blood. This is a protein called rhodopsin, which is in the membrane of the retinal cells in your eyes, and it's an exquisite light detector, and without it you wouldn't be able to see anything. So as you can see, proteins have many different shapes and do a variety of functions. Now, proteins are made using instructions in our genes, and so we all are familiar with the idea that different organisms have different numbers of genes, and it came as a shock that humans, actually, we don't even have 35,000 genes. It's revised downward. And we don't have any more genes than the lowly worm, C. elegans. So, it puts that into some perspective. And so the question is, how do you get from genes to proteins? Well, genes are units of information that reside on DNA, which, you all know about this double helical structure, and how there are 4 building blocks that make up a DNA chain, and the opposite strand has the 4, but they match each other or pair with each other in this complementary way, so that if you know the order of these building blocks on one chain, it automatically specifies the order on the other chain. And that's how, when 2 strands of DNA separate, each contains the information to make the other strand. And that essentially was what Watson and Crick called the secret of life, how you can get from 1 cell to 2 cells, or how you can pass information on to your offspring. But the problem is, protein and DNA chains are very different. So, as I said, DNA consists of 4 types of building blocks. And what happens in the cell is it's copied. A section of DNA that contains information on how to make a protein is copied into a temporary molecule called messenger RNA, which is very much like DNA, except it's just single-stranded rather than double-stranded. And from there, you have to get to a completely different type of polymer. So somehow the information here contains information on how to make a completely different type of polymer, which consists of 20 different building blocks. And the way that's done is that you go from the single-stranded DNA to the RNA molecule that I just described, and now you have to get to this completely different type of polymer, which is what a protein is. And the way that's done is that each group of 3 of these 4 types of building blocks is read as a unit called a codon. So you can imagine, if you only read one of them at a time, you'd only be able to specify 4 types of amino acid building blocks, but there are 20 amino acids which make up proteins. So if you read them 2 at a time, you'd have 4 times 4, or 16 possibilities. it was still not quite enough. But if you had read them 3 at a time, then you'd have more than enough possibilities. And so that's how it's read. But the way it's read is that there's an adapter molecule, which at one end brings along the amino acid to add to a growing protein chain, and at the other end has 3 bases that essentially recognize the bases on RNA by pairing in just the same way that the bases can pair across the 2 strands of DNA. It's the exact same chemical phenomenon. And then a second tRNA can read the next group of 3 and bring a new amino acid and so on. Now, this process doesn't happen by itself. It's not as if the RNA can lie around and this tRNAs can just assemble on itself. It actually turned out that cell biologists were looking at where proteins were made inside cells. And by radioactive labeling, they found out that newly made proteins ended up in these particles on an organelle called the endoplasmic reticulum in our cells. And when they isolated these particles, they found out that they all were about the same size, and they all seemed to consist of 2 parts, a large and a small part, which are called subunits, a large and small subunit. And because they were isolated from microsomes, these are essentially fragments of the endoplasmic reticulum, and when they analyzed them, they were known to be made of both RNA and proteins. So they were called the ribonucleoprotein particles of the microsomal fraction. Now, at a conference in the late 50s, people got tired of saying the same phrase over and over again. So somebody named Howard Dintzes, who's an emeritus professor at Johns Hopkins, said, why don't we just call it the ribosome? And that's the name that has stuck ever since. People went on to analyze what ribosomes were made of, and they found out that in bacteria, there were 2 subunits which consisted of large pieces of RNA, along with about 50 different proteins. And these formed the large and small subunit, which then assembled into the whole ribosome. And if you looked at higher organisms, there were about 80 proteins instead of 50, so they were over half again as big, and the RNAs were bigger as well. And so the ribosomes from higher organisms are much larger and more complex, but the core of the ribosome, I should say, is very similar between bacteria and humans. So to give you a schematic idea of how the ribosome might work, it's the small subunit that binds the genetic message, the mRNA, the messenger RNA that contains a piece of the gene. And the first tRNA then comes in and binds, it has these slots, I should say, has these slots for the binding of those tRNA molecules, which are those adapter molecules that recognize those units of 3 bases on the genetic message at one end and bring in the amino acid at the other end. And at the beginning, the first tRNA binds in what you might call the central slot. And then the next tRNA, which correctly matches this next group of 3 bases, binds to the ribosome. If it doesn't match, the tRNA leaves. But if it matches, then it's accepted, and then the ribosome joins up the first and second amino acid. And then it has to move along the mRNA, and that happens in 2 steps. And then the old tRNA is kicked out, and you can see that the new tRNA has now moved into the central slot. And then the next tRNA comes in, and then amino acids 2 and 3 are joined. So you can see what's happening. The ribosome is reading the message here, and according to the codons and what amino acids they specify, it's accepting just those correct tRNAs that will result in synthesizing the right order of amino acids on the protein. So it's synthesizing the protein in accordance to the instructions, accordance with the instructions from the gene. Now that was a very simplified cartoon, and in the mid-90s, people had a more realistic idea of what the ribosome looked like by electron microscopy, and this is what it looked like at the time. And you can see that the genetic message, it's not straight, but it actually snakes around a cleft in the small subunit. And these tRNAs are not little stubby things, but they're actually L-shaped molecules, which at one end recognize the codons, and at the other end bring in the amino acids. And in the central slot, the tRNA holds the growing protein chain, and the new amino acid brings in the new tRNA there. So if you want to, that's fine at one level. You can understand quite a bit about the ribosome. It's really a tribute to biochemists and geneticists that they'd figured out all this without actually knowing what it looked like. But if you really want to understand the chemistry of how this works, this is a complicated machine that has to accept things, synthesize things, and also move along through the whole process. And then you want to understand how antibiotics block this machine. Then you really need to understand its structure. And the problem with visualizing small molecules, atomic details, is that you can't do it using a light microscope. And to give you, to show you what you could do with a light microscope, the way we normally visualize small objects is you would take an object and you would use a lens. And what the lens does is gather scattered rays from the object and combine them into an image. And in fact, all of you, if you're looking at the screen, you're actually looking at scattered rays from the screen that the lens in your eye is combining into an image on your retina. The image is much smaller than the actual object on the screen. But of course, if you were to reverse it, you can also have a microscope where the image is much larger than the original object. And that's how people look at very small objects. But the problem, as I said, is that if you want to look at atomic detail, you cannot use light, because Ernst Abbe, a 19th century physicist, showed that you can never resolve two objects that are closer than half of the wavelength of the light you're using. The wavelength of light is about 500 nanometers or 5,000 angstroms. The distance between atoms is only about 2 angstroms. So there's no way that you could get any sort of atomic structure by using light. You could use electrons, but at the time this all happened, electron microscopy had not developed to the point where you could do that on biological specimens, although physicists and material scientists could have used electrons and an electron microscope. So for the 100 years from about 1912, the only technique that could give you a direct visualization at the atomic level was a technique called X-ray crystallography, for which Cambridge has a large tradition. It was, the idea of it to solve structures was developed by Bragg when he was a PhD student in Cambridge and became the youngest ever science Nobel Laureate, which still remains. And the use of it for proteins and large molecules was pioneered by Max Perutz and John Kendrew, who founded the institute where I work, the LMB. And the way it works in a very simple way is that you have to make crystals of the object, and then when you hit it with X-rays, because the crystal is periodic, there's interference so that the scattered rays, instead of going all over the place, for example, the scattered rays from the screen are going all over the room, so no matter where you sit, you're seeing scattered rays from the screen. But if you have a crystal, the scattered rays are enforced along certain directions, which gives rise to spots. And they sort of look like that. So here's a typical diffraction pattern from a crystal. But the principle is the same, is that if you somehow have these scattered rays, and you could do in a computer what a lens does in actuality, which is to calculate a Fourier transform, then you could create an image, you could calculate an image. And because there isn't really a lens for X-rays, but you could do it in a computer. You could do exactly what a lens does, which is recombine the waves to calculate the image. So you could do that, and then you would get an image. Now, of course, you know, there is a slight problem with this. So I like to say you can join a ribosome lab and see the world, because ribosome crystals are intrinsically very weakly diffracting. So they need these extremely powerful sources of X-rays, which are synchrotrons. And these are some of the synchrotrons that my group had used in order to solve the structure. Where, of course, this is actually a lie. You know, the US Navy used to have a famous ad, join the US Navy and see the world. If you signed up for it, you would only see the inside of some horrible battleship. You wouldn't even actually be seeing much of the world. And of course, my group, they don't actually go skiing in Grenoble. They actually, you know, or in Switzerland. This is what they're mostly seeing, which is seeing the inside of an instrument at the synchrotron. And this is the tube that brings in the X-rays. The crystal is here. And this is a CCD detector. We now have better detectors for X-rays, which actually measures the diffraction pattern. Now, if all of your experiments have worked well, what you end up with is an image of your object. You don't end up with a chemical structure of your object. Rather, you end up with a three-dimensional image. Now, this three-dimensional image has to be interpreted in terms of a chemical structure. And that's like solving a complicated jigsaw puzzle, but it has some real differences with our usual jigsaw puzzle. One is that it's in 3 dimensions rather than in 2. A second is, in a real experiment, there are errors in the data. So there will be pieces that show up as image that are actually just noise and don't belong to the image at all. And there are other pieces that are missing, because those parts of the molecule are you know, not fixed. They're mobile, and so they're blurred out. And so you have to be careful about that. You'll be missing certain things, and other things don't really belong in your image. And then, of course, the most important difference is that you aren't conveniently supplied the answer on the cover of the box, okay, because you have no idea what this molecule looks like. But in other respects, it's very similar. Just those of you who've done jigsaw puzzles know that you start off with parts that you can interpret very easily. For example, the edges, and then other parts which have a very regular pattern. And then you leave things like grass and leaves and sky for the very end. And that's sort of what you do here. So you look around, and I'm afraid this is a little... well, with the light, it's hard to see, but maybe it'll be clearer here. That's right. So what you see there are 2 ridges that you can see. So these are 2 strands, and if you know what you're looking at, you can interpret these as a double helical strand of RNA. RNA can also form double helices. And you can see these are the sugars, the phosphates that alternate and form these little bumps. And you can see that they're interacting with each other, and those are the formation of base pairs between the 2 strands of RNA. So now, all of a sudden, what you've done is you've taken a piece of this, you know, image, and you've interpreted it in terms of a chemical structure. And so you've got a piece of the molecule now interpreted in atomic terms, and you can then do that for the rest of the molecule. And when you do that, what you do is you just keep on interpreting as much of the image as you can, and you keep building away in molecular terms until you've effectively got nothing else to account for. And then you basically declare that you've solved the structure, and send it off to be published. So using exactly that, the large and small subunits of the ribosome were solved in 2000. And one of the things they did indicate was, the ribosome is the object that makes proteins, but it itself is made up of both RNA and proteins. So if it itself consists of proteins, where did the ribosome even come from? And Francis Crick first had the idea that maybe early primordial ribosomes didn't have any proteins and consisted only of RNA. And maybe today's proteins sort of evolved from a primordial ribosome. And the structure supports that in very strong terms, because if you look at the active site pockets where the chemical reaction to join the amino acids occur over here in the large subunit, or where the mRNA binds or the tRNAs recognize codons, they consist almost entirely of RNA. And that means the ancient core that was involved in these activities consisted only of RNA, and maybe the proteins got added later as it kept making proteins, some of them bound to itself to make it more efficient, and that drove its selection. So, that's one thing that came out. Now, it took us another 6 years to get a structure of the entire ribosome with mRNA and tRNAs bound. At the time, it was the largest structure solved by crystallography. It consisted of over 1 million atoms. And so it's astonishing that the method could actually do something like that. Now, solving the initial structure was obviously quite hard, and was a long journey that started with the discovery of crystals by Ada Yonat, and then many years before progress towards a high-resolution structure. But once the structure was solved, it was possible to rather quickly solve the structure of antibiotics bound to the ribosome. And that's because once you have the crystal, you can soak in antibiotics, redo the experiment, and do a difference. And the difference tells you where the antibiotic is. And so, for example, this little blob here shows you where the antibiotic spectrenomycin is. And if you can see that it can fit spectrenomycin, we can precisely orient it. And here it is in the structure of the entire large subunit. So this is only a few hundred atoms, a few hundred Daltons, maybe a dozen or so atoms. And it's in a structure of over 200,000 atoms. And so, yet you can actually accurately determine its precise binding site and its orientation when bound to the ribosome. And so, if you then look at where it is on the in the small subunit, we determine the structure of a number of antibiotics bound to the small subunit. Now, at the same time, others, notably the group at Yale, were determining the structures of antibiotics bound to the large subunit. And so, for example, here in the large subunit is the pocket where the amino acids are joined together when a protein chain is being synthesized. And then the protein chain has to emerge through a tunnel in the large subunit. And there are antibiotics that bind both at the entrance to that tunnel as well as at the site where the amino acids are being joined. And just to give you an idea, here's the ribosome, and here are the 3 tRNAs. This is the tRNA in the central slot that either holds the first amino acid or the growing protein chain. And in green is the new amino acid that brings in the new tRNA that brings in the new amino acid to be added. Now I'm going to zoom into the site where the amino acids are going to be joined. And if you sort of just follow the movie along, what you see is here where the 2 amino acids are that are going to be joined, okay? This is the new amino acid, and that's the first one. And one of the antibiotics, chloramphenicol, binds right here, okay? Now, chloramphenicol is a very effective antibiotic, but it's also quite toxic. And for that reason, it's not used clinically as often as it used to be a while ago. For example, one of the side effects is anemia. And I may be one of the few people who's had chloramphenicol in this audience because it was used to treat typhoid, in the 50s and 60s. Now, here's another class of antibiotics called erythromycin, which binds at the mouth to the tunnel. And you can see how it's very close to the chloramphenicol site. Now, here are the two antibiotics. There's chloramphenicol. There is, azithromycin or erythromycin, the macrolide antibiotics. Erythromycin is quite a good antibiotic, but a number of strains of bacteria are resistant to macrolides, to erythromycin classes of antibiotics because of a mutation in the ribosome itself or other things. Chloramphenicol is very effective, but it's also toxic, okay? So what happened after these structures came out is that pharmaceutical companies became interested in using these structures to develop new antibiotics. And one idea that a company that used to be called Ribex, but that's now called Milenta, that Tom Stites, one of the people I shared the Nobel Prize with, founded in New Haven, was they said, well, we've got these two antibiotics next to each other. What if we make an antibiotic that spans both sites? Then it could be highly specific, which might reduce its toxicity, but it'd also be highly effective. So they synthesized a compound that they called RX-2102. And then, of course, you can make a compound and predict computationally that it ought to bind, but you're never sure until you actually do the experiment. But when they did the experiment, they found indeed it bound as they might have predicted. And this compound, at least in assays in the lab, was very, very effective against a variety of strains, okay, that are resistant to macrolides. So now you have a compound that's very effective against resistant bacteria. Of course, to go from a compound that's effective against resistant bacteria to something you can inject or give to a patient is a long road, because you don't know what the properties of, first of all, you don't know how cheap this is to make. You don't know its toxicity. You don't know whether it sticks around long enough, whether it enters the bloodstream. There are huge numbers of problems. And only a small number of lead compounds actually make it into drugs. And that's why, before I go on to that, there are, of course, many other possibilities, because there are many other natural antibiotics that bind next to each other. So you could play this game in many ways. So that potentially you could use these studies to do lots of things. So it takes a long time to actually develop a drug. But the point is that even if you actually have a drug, it's inevitable that you will get resistance. And the reason is that bacteria have evolved multiple strategies to acquire resistance to antibiotics. The way antibiotics or any drug works is by a tight fit between the drug and the pocket in its target molecule. So if it's an antibiotic that binds ribosomes, it'll find a pocket in the ribosome and it binds tightly to that pocket. Now you can destroy that tight fit in a variety of ways. For example, you could have enzymes in bacteria that cleave the antibiotic. So it's cleaved into at least two pieces, and each of the pieces doesn't fit very tightly in the pocket. So the affinity isn't very high, and so it doesn't bind it well anymore. And that's how penicillin resistance occurs, by cleaving the penicillin molecule, so it no longer works. The other common strategy is bacteria have enzymes that actually add extra groups to the antibiotics. And what this does is it changes the shape of the antibiotic molecule, so it no longer fits in the pocket properly. A third strategy is that it mutates the target molecule itself, so the pocket itself is changed. So it no longer has the same chemical nature, and so the antibiotic no longer likes to bind in that pocket. That's another mechanism. And finally, another mechanism is the cell has pumps, which have evolved to pump out things the bacteria doesn't want inside the cell. And either fortuitously or by evolution, these pumps can now pump out lots of these antibiotics. And so that's another mechanism. So there are multiple ways that bacteria can acquire resistance. And the only, I think, given the power of evolution and natural selection, probably the only thing we can be sure of is that when you make a new bacterium, a new antibiotic, eventually resistance will occur. And so the question is, what are the difficulties in developing new antibiotics? Well, the first is the failure of the profit motive. And that is, I told you, getting a compound is one thing, but making sure it's a safe and efficacious drug is quite a different thing. And so it costs about $1 billion to bring a drug to market from an idea or a compound. Now, many drug companies have actually given up on antibiotic development. And that's because if you have a new antibiotic, you want to restrict its use only to patients who have resistant infections, because you don't want to spread this antibiotic around. Otherwise, you're going to develop resistance to the new antibiotic. And so the patient pool is very small. And also, if the antibiotic is effective, the patient is cured, which is not a good thing for the drug company, because they're no longer a customer at that point. I joke that the ideal customer for a drug company is, a diabetic with high cholesterol and possibly impotence as well. And so they're in the market for, pills for the rest of their lives. And so, so that is a real problem with antibiotics. And so the result is almost no new class of antibiotics have been developed in the last 2 or 3 decades. And this company that was founded by Tom Stites actually tried to launch an IPO a few years ago, and the share price it commanded was such that they withdrew the IPO, and then it eventually was bought by somebody else. And oddly enough, I think within a month of that, a company launched an IPO successfully for many billions of, I think hundreds of billions of dollars. And the main product of this company was to be able to share gossip and photographs and so on, and as we've learned more recently, fake news of all types. And so, there you have it in a nutshell. A product that could save lives, isn't worth anything, and or isn't worth what we ought to value it at. So I think we need a different model. So we need alternatives to the free market. And it's well to remember that the first sort of modern antibiotic, apart from sulfa drugs, was penicillin. Now, penicillin was developed by a huge government, initiative. It was the British government in response to soldiers dying in World War II that launched a massive effort to produce penicillin. So we need governments. We also need multinational agencies, possibly like, the WHO or, Gates Foundation and so on, and charities. So, of course, new antibiotics are only one part of the equation. Really, if you want to combat antimicrobial resistance, you're going to need a coordinated strategy. And the first is you need good surveillance so that you know when resistant bacterial infections occur. You also need infection control, for example, public sanitation and hygiene. Even simple measures like washing hands have dramatically cut down on the rate of infections in hospitals. And isolation of infected patients is another thing. You also need, when you have antibiotics, you need to promote rational use. And for example, especially in the US, there have been thousands, I think 2 million as prescriptions for the viral infections every year. And these are antibiotics are absolutely useless against, common cold or the flu. Another thing is unnecessary prophylactic use, where you just take it, because you think you might get something. And although in some cases it's required, if you're immunosuppressed or something. And a really particularly bad use of antibiotics is its use in agriculture. It was fortuitously discovered that if you gave cattle antibiotics in their feed, they would grow fatter and bigger. And since then, many farmers all over the world are using antibiotics in cattle feed, including, I should say, the EU, which prides itself on its, sort of health and safety regulations. And so I think that is another thing that we can do. And of course, scientists can contribute, and we can contribute in various ways. We can understand how bacteria cause disease, because that's not even long after the bacterial agent of a disease is known, the mechanism by which it causes disease is not really well understood, and that's complicated cell biology and host pathogen interactions and so on. And that, if you understand those mechanisms, you might also have new targets. Another useful thing that is happening is rapid diagnostics, and especially cheap kits to identify infections very quickly, so that you give antibiotics only when they're appropriate, and you can also select then specific antibiotics for that particular disease or bacterium. The development of vaccines is a long-standing problem, and I think people still don't understand why some vaccines are easier to make and others, are not effective. And what I talked about today was about new drugs and therapy, which is synthesizing how compounds can block a target and how you can do that. So I finally want to close by just showing you a movie of the ribosome, because after all, you need to sort of at least see what a grand machine this is. And so this is the small subunit, and it recognizes where to start on the genetic message, and that brings in blue is the first tRNA molecule with the first amino acid. And at some point, the large subunit joins, and then these proteins that help the ribosome start at the right place leave. And when they leave, the ribosome has the first tRNA with the first amino acid, and it's ready to start accepting the next tRNA. And that's the beginning of the elongation chain. And so the next tRNA is brought into the ribosome, and it's brought in by protein factor. And if it's the correct tRNA, the protein factor lets go so that the tRNA can then swing in to the pocket in the large subunit over here where the amino acids are joined. And then the tRNAs have to move, and that happens in 2 steps. First, the tRNAs move with respect to the large subunit, and then another protein factor helps to nudge them along so that the tRNAs and mRNA move with respect to the small subunit. And then now the whole assembly is shifted over by 1, so then the next tRNA can come in, and then this process continues, and you can see how different parts of the ribosome move. Look like this gate moves to let the tRNAs out, and the whole tRNAs, the ribosomes ratchet, and the growing protein chain emerges through this tunnel in the large subunit and folds up into a 3-dimensional shape, which is characteristic for each protein has its own characteristic structure. And this continues until the ribosome reaches the end of the coding information. And that's a special codon called a stop codon, which is recognized by a protein, which recognizes it and cleaves off the newly made protein so it can go off and do its thing. And then another factor comes along and splits the ribosome apart so that the whole process can start over again. And while you've been listening to me, the thousands of ribosomes in the trillions of your cells have been busily making tens of thousands of protein molecules in each of your cells. So thank you very much.